and I look forward to hearing more.
Yeah. Thanks, Keon and Wendy. I'll introduce myself for whoever joins just now. My name is Richard Martinez, and I'm a graduate student in Elena Niño's Honeybee Lab here at UC Davis. And today I'll be talking to you about honeybee nutrition, from fundamentals to recent findings involving, and I'll be sharing updates from my own research in honeybee nutrition, which revolves around dietary supplements. So let me turn off my camera briefly. I can figure out how to do that. Okay, cool. So here we go. So here's an outline of what I'll be covering in this talk. I'll start by covering why we need research into honeybee nutrition. And next, I'm going to provide you with the background on honeybee nutritional requirements and their roles. Next, I'm going to cover contemporary issues with nutrition, something we live with today, and followed by scientific approaches to nutritional additives. And lastly, some final remarks. So the need to research honeybee nutrition. Honeybees are important agricultural pollinators. Like, they're responsible for approximately $15 billion worth of produce in the United States. Sorry. Ensuring the health of honeybees is a top priority for researchers, the agricultural industry, and beekeepers alike. There's so many factors influencing honeybee health. It includes biological and environmental stressors, as well as the impact of these stressors on the individual bee and the colony. The image on screen depicts the dynamic relationship these stressors have with one another, as well as with the honeybee. Honeybee health has been declining for decades, and some factors driving these losses are pesticides, pests, pathogens, and poor nutrition. Earlier this year, a survey was conducted by Project APSM, the American Beekeeping Federation, and Honeybee, the American Honey Producers Association. The survey was covered by Bee Culture magazine, and the survey estimates that the commercial beekeepers lost an average of 62% of their colonies between June of last year and February of this year. And just a few weeks ago, in the press release from Washington State University, entomologists predict that colony losses could reach up to 70%. To put this in perspective, the average loss in the past 10 years has been between 40 and 50%. Now, some background on honeybee nutritional requirements, roles, and behavior. What do honeybees need? Well, carbohydrates, proteins, lipids, which includes minerals, vitamins, micronutrients, and of course, water. Where do bees get these resources? How much do they need? Well, nectar and pollen provide the brunt of carbohydrates and proteins. This was discovered in the 80s. And for adult workers, their sugar intake revolves around 4 milligrams per day to survive. But for the larva, the type of diet must change for successful rearing. It increases in sugar concentration towards the last 2 days. Also, the acidity of bee bread shifts and becomes less starchy. Gut bacteria helps metabolize a lot of the compounds. It helps break them down and makes them more nutritious for honeybees. And lastly, again, the larval diet transitions from amino acids to carbs. So what happens if bees don't have proper nutrition? So it can lead to reduced brood rearing or less amount of brood rearing. There's some research to support the conclusion that honeybees won't rear a bad bee. So if nurse bees detect that they're low on food supplies, but the queen has already laid eggs, those eggs will just dry up and die. They won't get fed. Some other impacts of poor nutrition include higher worker mortality. There's a lot of research that ties immunity response to nutrition. So if your bees are suffering from malnutrition, they're more likely to be affected by pathogens and pests. Uh, Poor nutrition also leads to a lack of interest in queen caring, which also would ultimately result in the demise, the death of the queen. So as I mentioned earlier, reduced immunity, stunted maturation of flight muscles. So when bees become old enough to forage, if they're suffering from malnutrition, they won't have those muscles strong enough to fly. Poor nutrition also impacts the development of hypopharyngeal glands in the ovaries, which I'll talk about later. And the impact of nutrition can be seen at a genetic level. In this case, vitellogenin, which is an important protein in egg development and bee development in general, is expressed lower in bees that are suffering from malnutrition. Now, who does the foraging? Well, foragers do. And foragers are roughly about 20 days old, but this is highly variable. And depending on the time of the year, it can be between 25-day-old bees to 45-day-old bees. So that shaking you see is known as the waggle dance, and it's language that informs other bees the distance and quality of the food source. Pollen is crucial for the bee's nutritional needs, but when it's lacking, their ability to signal food sources is compromised, and they're less likely to perform the waggle dance to recruit other bees to food sources when they're starving. So let's say a forager actually does make it to this food source. Well, then they forage nectar, they collect it, they bring it back to the hive. It goes through a few steps. Some enzymes are added and it's dried. This becomes food stores for winter and dearth. Carbohydrates in the form of honey also contribute to wax production. And a more recent discovery, which is roughly 10 years now old, is that nectar contains phytochemicals that are involved in development and pathogen resistance. I'll come back to that later as well. Now, when a bee forages for pollen and they bring it back, pollen meets the nutritional requirements for proteins, lipids, and micronutrients. But pollen also promotes reproduction in the queen. Pollen is also used in the manufacturing of brood food for nurse bees, and pollen has a positive immune response on foragers, as well as detoxification pathways in honeybees. And of course, bees also need water, not just to drink, but provide climate control in the hive. Bees also forage for plant resin, and they mix it with wax to produce propolis, a glue-like substance with antimicrobial properties, which aids in honeybee health. So here are some contemporary issues with nutrition. As I mentioned just now, water. California suffers from drought, and the frequency and severity has gone up over the years. Water is necessary for nurse bees to produce liquid food to feed larvae. On the screen, you see on the top left a normal fed brood, what normal fed brood should look like. It's moist, it's milky, it's opaque liquid. And on the lower right, you can see almost clear substance in low quantities. That's what water can do to your hive, or lack thereof. Another issue is monocultures. The agricultural setting is just not the best for meeting nutritional needs of honeybees. It's one crop, one profile of carbs and lipids, or not the adequate amount. And it's just at one go, there's not enough bees to pollinate all the flowers, let alone having to pollinate at one specific time of year. So, for example, the fact that almond pollination occurs late winter, but then forage diminishes throughout the year is an issue for honeybees. They need food throughout the year, not all at once. Another issue with monocultures is pesticide residues. Growers treat their crop with pesticides, and although honeybees might not be placed in the orchards at the time of the spray, there's residues on the flower which bees are exposed to when foraging. Another issue is urbanization. So due to housing developments or just what's it called? Yeah, just pretty much that. There's a reduction in plant diversity. So a lot of environmental pollutants also that honeybees or just pollinators in general aren't accustomed to in a natural setting are present in a city. Lastly, the last issue I'll cover is antibiotics. So honeybees treated with antibiotics for diseases show a decreased lifespan in altered gut microbiota. I mentioned earlier how gut microbiota is important in breaking down certain molecular compounds in the food. It promotes a healthy lifestyle for honeybees. Now, I'll cover some recent research regarding antibiotics, but if you do find yourself with the need to use an antibiotic, be aware of the side effects. Now, some scientific approaches to nutritional additives. I'm going to introduce y'all to a concept known as precision agriculture. So it's the implementation of tools across agricultural systems intended to provide growers with real-time data about the quality of their crops to drive decisions. It's tech in agriculture to drive decisions. That's what technology is used for in precision agriculture. So how can growers use this data? Well, a lot of this technology can determine what is if there's unequal fertilization on your crop, if there's water deficiency, if there's a pest presence, because a lot of pests they lead to leaves wilting, and this can actually be picked up by LiDAR. So it's specific technology to pick up different light waves. The reason why I bring up this topic is because there's a parallel to this in apiculture. It's called precision nutrition. Although it doesn't involve technology, its goal is to improve practices in search of a diet that compensates for or promotes certain aspects of honeybee fitness. So in the image on screen, you can see that on the outer ring, which is kind of a olive green color, that's what happens when there's good nutrition, right? Flora diversity promotes good nutrition. Key molecules are present. Abscisic acid as well is present. The inner ring, the pastel blue, shows what happens when there's low flora diversity and key molecules tend to vanish. And then all these bees go through the same dearth and winter environment and can come out the other side vastly different. Now, I'm sure you heard this before. Natural nutrition is the best. Yeah, of course, wild forage. But we are in a drought state. So foraging isn't always available for bees. So we got to pay attention to what's blooming and when. And if you have hives at home, just make sure they're bringing in food. There's also some resources available here on this link. In Northern California, however, we provide carbohydrates in the form of sugar water or sucrose solution between July and February. There's an example of sugar water that we use, a combination. And there's also various supplements you can use for carbs. So we have ProSweet, which is commercially available, high fructose corn syrup, and fondant as well. Keep an eye on California buckeye because it is toxic to honeybees. I'm going to cover now how we feed. So there's different ways to feed to supply your colonies with carbohydrate supplements. We got feeding cans, entrance feeders, in-hive feeders, as well as top feeders. Now for protein supplements, you can buy pollen patties. Now there's a variety of pollen patties out there. It's up to you to decide to weigh the odds, the pros and cons of some of these. But and they come in a variety of dry and wet. And like I mentioned earlier, pollen is important for brood development. Now, if you're gonna store a sucrose solution or if you're gonna heat it up, you might want to pay attention to this. When sugar is heated up to high temperatures, it produces this compound on screen called hydroxymethylfurfural, or HMF. HMF is potential carcinogenic in humans, let alone honeybees. It exists naturally by the natural breakdown of sugar at high heat, but there's also other things that can impact the production of HMF. For example, the pH, so the acidity of the container you're storing it. Storage temperature, if you're going to keep this long term, that also plays a role in HMF production, and as well as water content. If your container isn't dry, you run the risk of producing HMF faster. And HMF alone is toxic to honeybees. You need a high concentration for it though. But given the current state of honeybee health, it's reasonable to suspect that that tolerance has decreased. So a bee that is suffering from poor nutrition will probably be more susceptible to dying of HMF poisoning if it's present at lower concentrations. Now, HMF usually only develops or breaks down if you heat sugar up to a temperature of 250 degrees Celsius. So pretty high temperature. And it's very unlikely for you to produce that if you leave a can of sugar out in the sun for like a day. You will produce it, but not at large quantities where it becomes toxic. But you also, again, have to mention the resistance, the tolerance for HMF is probably lower in honeybees now. In a study done with larvae, where larvae was fed HMF, you can see on screen, there's a survival curve. So on the x-axis, we have the days or times in this case. And on the y-axis, the vertical line, we have survival probability from 100 to 0. And we find that this green line, that shows a precipitous drop in survival is attributed to concentration of HMF at 5,000 parts per million. This bold solid line is a concentration of HMF at 10,000 parts per million. And then there's this one hidden in between, purple dotted line, refers to the 7,500 parts per million concentration. So this shows that if you feed larvae HMF, and if HMF is present in your hive, the larva will die. And this was done, what 10 years ago now? So be cognizant of how long you store your sugar water or your carbohydrate supplement. And I recommend not heating it up to excessive temperatures. Warm water, if you're mixing it with water, is sufficient enough. And I know you want that slippery, that you want that sugar water to spill out easily so you can feed it rather easily instead of that thick, viscous substance. But again, I digress. HMF, be aware, be careful. Now, so now I'm going to shift to another dietary supplement, probiotics. So probiotics in humans promotes gut health. It's the same for honeybees. There's a lot of products available, but around like 10 years ago, we weren't sure. Guess what? Now we are. So this is some recent updates on if probiotics help. So this study on screen. It was a research group out of South Korea where they were trying to test probiotic additives on pollen substitutes and see how that if that benefits the honeybee. What they found was this. So let me walk you through this real fast. On the horizontal axis, you have the different diets they fed their bees. And on the y-axis, you have the abundance of specific gut bacteria. And they're piled on top of each other. So it's a total of 100%, and whatever parts are shaded results to how abundant that bacteria is. I want to point out the biggest bacterial colony that was detected is Lactobacillus, which is a common genus in honeybees. So this is the control. I'm signaling here on the screen, and other diets showed an increased expression of this bacteria. So that's good. But diets 1, 2, 3, and 4 had that probiotic added to them. So you can see the difference between the control and the 4 probiotic added diets. Lactobacillus jumped like crazy. It increased by a lot, actually. And lactobacillus is actually one of the best, if not the most, like helpful bacteria in honeybees. It was followed by, let me see, bifidobacterium, which is the purple here. And in the control setting, you can see this thin purple line. That's the amount of that bacteria in regular honeybees. Now, if you add these probiotics, you feed them these probiotics, that bacteria also increases. And this is also a very beneficial bacteria. So how can gut bacteria help? Well, in recent studies, or 10 years now, but some studies showed that feeding a specific bacterial cell resulted in increased brood production and adult bee population, as well as honey storage. A different species of bacterial spores led to an increased brood production, adult population, and honey storage as well. And lastly, a probiotic product showed that it reduced the amount of Nosema ceranae spores numbers in infected honeybees. So probiotics, when used properly, are successful. They can provide an increase in growth in beneficial bacteria. However, just last year, researchers at the USDA found that probiotics were not helpful in improving gut dysbiosis, which is gut death, in bees treated with two antibiotics. The antibiotics they used were oxytetracycline and tylosin. But these researchers argued that the products they were testing had gut bacteria that is different from the gut bacteria honeybees have. So the product was introducing foreign gut bacteria that doesn't even exist in honeybees. So the reason why I bring up probiotic use is just to exercise caution when considering probiotic supplements. Make sure that the bacteria in the product correlates well with what we know honeybees having. So next I'll be addressing algae supplements. So algae is a eukaryotic cell. It's either a single or multicellular organism that can perform photosynthesis. Photos. Um, Algae is not a plant, it's not an animal, it's not a fungi, and it's not a bacteria. There it's its own classification, and they're called protists. And in recent years, scientists at the USDA have studied feeding algae supplements to colonies. And what they found was that a specific strain of algae, it increased egg laying capacity. Another strain of algae increased brood production. It also increased colony population size in the same study. More recently, algae appears to provide essential amino acids in levels equal to or exceeding that of other pollen substitutes. So it might just be a better supplement altogether. Also, a study showed that algae supplements increased survival when colonies were fed algal supplements, and that the brood areas were larger in colonies that were fed algae supplements, specifically spirulina, which is a specific species. And also it increased survival. And now, if you see algae in your nearby lake, I strongly encourage you to not collect it and do not attempt to feed your colonies this. The researchers used specific species of algae that yielded these results. There are some algae supplements that are commercially available, so I encourage you to weigh your odds, pros and cons. Now, I'm going to shift to a topic, supplemental diet, that I am somewhat familiar with. I'm very familiar with, and that's phytochemical diet supplementation. So what are phytochemicals? Well, they're compounds that plants produce to protect themselves from being eaten, UV radiation, and attract pollinators. These compounds are available to bees via the nectar and pollen, and they have positive effects on honeybee health. They can lead, so enhancing honeybee diets with phytochemicals has benefits. For example, it improves pathogen tolerance, it improved pesticide tolerance, it increased gut microbiome abundance, which I just covered, and it's a good thing, and it also increases longevity. Now, the compounds I used in my study were caffeine and p-coumaric acid. So my thesis revolves around feeding these two compounds to colonies and testing colony performance. So I fed my colonies a sucrose solution with one of the following chemicals, phytochemicals. Using an internal feeder, I provided my colonies one of four diets. It was the control, which is just sugar water, or sugar water with caffeine, or sugar water plus picomeric acid, or a fourth diet where it was sugar water plus both caffeine and picomeric acid. So I fed my colonies 4 consecutive weeks and then 4 more times if needed. Some of the data I collected from my project includes survival of colonies over a 5 month period, colony strength, which I measured using frame of bees. This is a measurement used in the beekeeping industry to measure colony strength. And then I did my own detailed assessments, which measured the average coverage of all these factors: brood, honey, nectar, pollen. And lastly, I tracked varroa mite density to see if my diets played a role in reducing varroa mite load. Now, I'm going to show you what I found. So on the horizontal axis, you have the date. So we started in, I started in late June and I went all through early December. And on the Y axis, I have the survival probability. So of these 4 diets, I found that the colonies fed the mixture of phytochemical solution had the highest survival. So we see here that at the beginning of December, mixture had about 60% of the colony survive, while the control and picomeric and caffeine had about 30% or less. Now, these colonies still die, of course, for the mixture. But as a beekeeper who has thousands of hives, would you rather take a 30% loss or a 60% loss? Food for thought there. Another data point that we found, let me show you. So here on the Y-axis, I covered the average brood found in colonies. And on the X-axis, I have the dates as well. Over a 5 month period, I found that the colonies fed the picomeric acid phytochemical solution had the lowest average brood content. So that's this orange line here. Which is an interesting thing to note, given that there is some background research that supports this really. And I'll cover that in a bit. But it's interesting that picomeric acid, I was hoping it would recover 3 months in, but just, yeah, didn't go so well. And for the other results, for honey, nectar, pollen, frames of bees, and varroa mite density, the diets had no impact on these metrics whatsoever. So I paired this colony trial with a lab trial where I collected bees that I controlled for it. And I controlled them in this manner. So I introduced an empty frame into the colony that I wanted from the first experiment. I let the queen lay eggs on it, and then I let the nurse bees feed these eggs the corresponding diet of those colonies. But before they emerged, I removed the frame when the cells were already capped, and I let them emerge in an incubator. After emergence, I placed them into plexiglass cages you see on screen, and these bees were presumably reared on these diets, which is an important distinction to make. After emergence, I fed them a regular sugar solution, no additives, and I tracked how long they lived for. Now, previous studies involving cage bees only fed the bees the experimental diet after their emergence. These bees were presumably were fed the experimental diet during development, so there's a difference. Now, the question is, can rearing Can rearing bees on the phytochemical solution affect survival of those bees? I found that, yeah, rearing bees on picomeric acid had a large impact on the survival of those bees in the long term. Now I'm going to summarize what I just spoke about briefly. So, in summary, the mixture of phytochemicals led to higher survival in colonies. Picomeric acid appears to decrease brood production temporarily, and honeybees reared on phytochemical diets, specifically picomeric acid, lived longer. And here's the background I was talking about earlier. Not all phytochemical effects are positive. Some not so great effects are that the phytochemicals that I used can lead to a different size hypopharyngeal gland, which is what HPG stands for, hypopharyngeal gland. Hypopharyngeal glands are organs in the back of the honeybee head that play a role in producing royal jelly and food for honeybees. So, as you see on screen, that picomeric in previous studies, picomeric acid led to a larger hypopharyngeal gland, and caffeine led to a smaller hypopharyngeal gland. So the size of the hypopharyngeal gland indicate what task that particular bee is supposed to be doing. It also gives us an idea of how old it is. Some implications of these results involve the tasks the bees in the colonies carry out. For example, if a newly emerged bee is fed caffeine, they may transition to a forager role, jumping over their nurse bee tasks, potentially hindering brood production because now all your nurse bees are no longer nurses, they're foragers. That's what happens with caffeine. It shrinks the hypopharyngeal gland. Now, let's say you have older bees and you feed them picomeric acid. Picomeric acid leads to a larger hypopharyngeal gland, which is indicative of nurse bees. So these older bees might regress to their previous role of brood rearing, which now impacts the foraging force. So some future directions for supplemental diets is to explore how picomeric acid impacts queen egg laying ability, since we noticed on my project that brood production decreased. But also, can we rear better queens if we feed the hive these supplemental diets? Not just the phytochemicals, I mean, all the algae, the probiotics, everything. Can we rear better queens? Now, I'm just gonna do some final remarks, just a summary of pretty much all I've spoken about. So carbs, carbohydrates, proteins, lipids, vitamins, and minerals are all factors that drive colony growth, survival, and strengthens immunity. Poor nutrition has consequences down to the genetic level, and dietary supplements appear to counteract some negative effects of poor diets. And this includes phytochemicals, this includes algae supplements, as well as probiotics. Now, if after this presentation you are motivated, you're pumped up, and you want to take action to protect honeybees and our food systems, here's some things you can do. Plant native forage. Can't stress this enough. Honeybees need diversity in pollen sources, better nutrition. It leads to an improved immune system, an improved detoxification response, and it helps deal with pathogens and pesticides. Also, pollination. The pictures I've chosen for this slide were specific ones because these plants on screen provide the phytochemicals that I studied. So caffeine is found in citrus, as well as other grapefruits, lemons, oranges, and pomelos, I think, as well as coffee plants. So if I'm pretty sure, yeah, my compounds aren't available in on the market. So I would recommend put a hive next to a citrus orchard. They'll get that caffeine, they might get that boost. I will disclaimer, um, the concentration of caffeine in the wild is very low, so you would need to the bees would have to consume quite a bit of citrus nectar. The other images on the screen, for example, we got California buckwheat on the top right, and then a variety of roses and clover on the bottom. All those plants contain picoumeric acid, which was the other compound I studied. And so what I'm suggesting is pollinate all these things, and you might get the results of colony survival that I observed in my project. So with that being said, many thanks to all the folks at the Bee Niño Lab, the lab assistants, or the lab techs at the USDA, solutions expert Joseph Tauser, as well as the support scientists Laura Druville and research associate Chris Mayak at both the Niño Lab and at the USDA. Many thanks to them, or else this wouldn't have happened, this project. And lastly, thank you all for listening. And if you have any questions, I'll check the chat first, but then I'm open to taking your questions.